Okay, so here's part two. Part two is the urea cycle. So remember that cells can't store amino acids that accumulate as a result of our protein degradation, right? And so we really have two options. Either we recycle those amino acids for protein synthesis, or we deaminate them. We take the nitrogen off and we remove that free nitrogen, that free ammonium ion, and we reuse the carbon skeleton. So what do we do with that ammonium ion? Well, we go and we make urea. So the urea cycle occurs in the liver. And this is how we're going to remove all that toxic nitrogen, right? We said we can't store it. It's not good for the cell. So urea, how do we make urea? Urea is really made from that ammonium ion when either glutamine or glutamate are deaminated, right? So remember, you're trying to think about those nitrogen-containing groups. The other way that we can get it is we can get it from aspartate. So aspartate over here comes from our glutamate and our oxaloacetate, right? And then we kick off that carbon skeleton of alpha-ketoglutarate. And so, so we can get it from there. So we're getting a nitrogen group here. We can get a nitrogen group here. Now, if you look at urea, urea has two nitrogen groups. So we've kind of accommodated for the different ways that you can get the nitrogen. But you also have a carbon and an oxygen. So in the carbonyl, the carbon atom comes from the conversion of CO2 to bicarbonate, right, from the citrate cycle. So this is right from the mitochondrial citrate cycle. And then, so that gives you your carbon right there. And then the last thing that you need is an oxygen. The oxygen atom comes from water that's made in the final reaction in the citrate cycle. And so we can track and account for all the atoms that are present inside of urea. So let's talk about how we transport those amino acids in order to get them to the liver, right? We want to get them to the liver mitochondria, right? So there are three kind of different places that we can get amino acids from. So we've kind of talked about this before, right? That the first one is that we're going to digest our dietary proteins and we're going to get amino acids that way. Then the other way that we can do this is we can get it from glutamine from our peripheral tissue, right? All of your muscles and all of your peripheral tissue are going to make that excess nitrogen in the form of glutamine, which is really from glutamate and the ammonium ion. But that is your glutamine synthetase enzyme, right? Okay. Then the third way that you can get it is from alanine. So from your skeletal muscle, when your skeletal muscle starts to break down, you're going to produce alanine there. And so these three different ways that we're going to make urea, what we actually need to look at is what are we actually producing? Well, we actually have to make two urea cycle precursors. And those two urea cycle precursors are carbomyl phosphate. So that's what's right here. And then the other one that we need in order to run the urea cycle is aspartate. So these are the two things that are going to enter the urea cycle to eventually form urea. So let's talk about that alanine real quick. So you kind of remember the Cori cycle, right? So during rigorous anaerobic exercise, we're going to degrade our glycogen, right? We're going to build up pyruvate. That pyruvate in the Cori cycle, this is different, but in the Cori cycle, right, we reduced it to lactate. We exported it into the bloodstream. Then in the bloodstream, we took that lactate to the liver. And then in the liver, we converted it to pyruvate to remake glucose, right? That was the Cori cycle. So at the same time that that's happening in the Cori cycle, muscle protein is also being degraded. And so if muscle protein is being degraded, that excess ammonia has to be removed. And it's removed in what's called the alanine glucose cycle, which is really similar to the Cori cycle. But it's just our way to get rid of that ammonia. So in this case, we take that pyruvate and we use it to convert it to alanine, right? And so we have our glutamate that comes from the excess amino acids from protein degradation. Excuse me. Ooh, Whoops. So we have our amino acids, right? From the degradation, we convert that to glutamate. We can kick out the carbon skeleton in alpha ketoglutarate. We convert glutamate to alanine. Alanine can now be transported in the bloodstream, can go to the liver. Then we use that alanine to go through to pyruvate, to gluconeogenesis, and now we're making glucose. So we have this renewable source of metabolic energy that allows us to replenish the glucose that we need when our muscles are exercising a bunch, which is a really cool way. So then the other thing is to not forget, okay, what happened to, that's making glucose, but what happened to the nitrogen? So in the nitrogen, we convert it to glutamate, then we go through the urea cycle and export it as a waste product. Okay, so the urea cycle, right? Remember that urea is made in the liver. It goes to the blood. It goes to the kidneys, right? It's concentrated and excreted in the urine, right? That's our kind of big pathway of where we're going. So the urea cycle is really 5 big reactions. And so what I'm going to do is I'm going to walk you through them. It's a really good animation in the book that talks you through it. And then here are like the steps in kind of in brief words, but I'm going to take you through the whole thing. So we really have 5 reactions, right? Total. We have 2 in the mitochondria and we have 3 in the cytosol. Okay. So we're actually going to use 3 ATP molecules, but we're actually going to make 4 cleavages, right? So we're going to go from 3 ATP to make 2 ADP and an AMP, right? Per urea made. So per urea made. Okay. So that's kind of just big picture to keep in mind. So we're going to zoom in. All right. So we're starting at number 1 right here. So the key regulatory enzyme is in the very beginning. It's going to be our carbomyl phosphate synthetase. So synthetase 1 is in the mitochondria, 2 is in the cytosol. So, all right. So we're going to form our first product, which is right here. Whoa. Carbomyl phosphate. So here's our first product. It's formed in the mitochondria, and this is formed in the ATP-dependent carbomyl phosphate synthetase 1. So what do we need? Well, in order to form carbomyl phosphate, we need the bicarbonate from the citrate cycle. We need ATP to drive the reaction, and we're going to need our nitrogen-containing group, right, from our deamination of glutamine and glutamate, right? So from that, we've made our carbomyl phosphate. Now that we have carbomyl phosphate, right, that's what we use some of our ATP to phosphorylate it, to activate it. So now our carbomyl phosphate is going to combine with ornithine. This one is a lot like the citrate cycle, oxaloacetate, right? You need a substrate in order to enter the urea cycle. So it's the same kind of cycle that you have to keep in mind. So we're going to combine carbomyl phosphate and ornithine, and we are going to make citrulline. And so that's ornithine transcarboxylase that is going to create the citrulline. Now the citrulline is in the mitochondria, right? We're still in the mitochondria. We're going to have to export it out of the mitochondria into the cytosol. Once we have it in the cytosol, what we're going to do is we're going to activate it with AMP. So really you need to activate this, activate with AMP. So it's kind of an intermediate form. And so then when that AMP is replaced by aspartate, what we're actually going to make is arganosuccinate. And so that aspartate displaces the AMP and allows for the addition of the second nitrogen containing group, right? Because aspartate has a nitrogen and our citrulline has a nitrogen. So now we're going to make an intermediate here that has 2 nitrogen groups in it, right? So this is done in the cytosol. So this is catalyzed by our arganosuccinate synthetase, which is in the cytosol. So it results in the addition of that second nitrogen group. And at the same time, what's going to drive this reaction is that we're making a pyrophosphate. And so that cleavage of the pyrophosphate is going to drive that reaction, but that's that additional bond that's being used. All right. The next reaction is, again, a cytosolic reaction where we're going to cleave off the excess carbon groups. And so we convert arginosuccinate into arginine. And when we do that, we're going to produce fumarate, right? So fumarate's a product of the urea cycle. So now fumarate has no nitrogens. Our arginine has 2 nitrogen containing groups. The arginine is converted then to urea. So here's our second product here. We make urea, and then we also make ornithine. And so ornithine's right, just like oxaloacetate in the citrate cycle, in that we then pump it into the mitochondria to be used as a substrate in future reactions, right? So our 2 big products are urea and fumarate. And we don't necessarily say that ornithine is a product because ornithine is reused. Really what you're making is urea and fumarate. So what's the rate limiting step of this, right? The rate limiting step is right here. It's carbomyl phosphate synthetase. This is the rate step, and it's the key regulator. Right there, and like I said before, there's a cytosolic version. They call it carbomyl phosphate synthetase 2, but that's really important in pyrimidine biosynthesis. So we're not going to talk about it right now, right? Okay, so go in, watch the animation. The animation's really great, and then here's our step-by-step listed out and our overall reaction. Okay, but it's basically everything we said before. So let's talk about this regulation, right? So we said the key reaction in urea synthesis, right, is that we're worried about that carbomyl phosphate synthetase 1. So what feeds into that reaction? So there's an intermediate that regulates that control mechanism, and that intermediate is N-acetylglutamate. N-acetylglutamate is a metabolite that signals that we have really, really high levels of glutamate. Right? So you're only going to make N-acetylglutamate if you have a ton of glutamate to start with. And so you have this enzyme called N-acetylglutamate synthetase that makes N-acetylglutamate, right? And it makes it from 2 sources. It makes it from acetyl-CoA and glutamate. And so really the glutamate and the arginine, right? So if I can zoom out a little bit, right? Our glutamate right here and our arginine here, these are 2 things that are going to control the activity of the N-acetylglutamate synthetase enzyme. And this is what is going to stimulate flux through the urea cycle. So if we have arginine as that urea cycle intermediate and we have glutamate, we're going to really increase the rate of carbomyl phosphate synthase. And so then you're going to drive this urea cycle reaction, right? So the other enzymes are required for urea synthesis, but those other enzymes are regulated at the level of gene expression, right? In response to the amount of protein in our diet or glucocorticoid hormone signaling. But really our regulation here is the N-acetylglutamate biomolecule that controls carbomyl phosphate synthetase 1. And that's the key enzyme in the urea cycle. Okay, next one, we're going to talk about the Krebs bicycle.